Hello, Tina de Las Llagas here, and welcome to What a Beautiful Life. Today's lecture on the macromolecules of the cell will focus on the proteins. We begin with a brief survey of its building blocks, the amino acids, then the basics on protein structure and the function of proteins. Each of the major kinds of biological macromolecules, such as proteins, nucleic acids, and polysaccharides, consists of relatively small number, say from 1 to 20, of repeating monomeric units. These polymers are synthesized by means of condensation reaction, in which activated monomers are linked together by the removal of water. Thus, it's also the reason why it's called dehydration synthesis. And once synthesized, the individual polymer molecules fold and coil spontaneously into a stable three-dimensional shape or structure. These folded molecules then associate with one another in a hierarchical manner to generate higher levels of structural complexity, usually without further input of energy or information. Proteins are a class of extremely important and ubiquitous macromolecules found in all organisms, and it occurs nearly everywhere in the cell. In fact, their importance is implied by their name, which comes from the Greek word proteos, which means first place. Whether we are talking about, say, the conversion of carbon dioxide to sugar during photosynthesis, or how oxygen is transported in the blood, the regulation of gene expression by transcription factors, from cell-to-cell communication, or even the motility of, say, a flagellated bacterium, we are dealing with the different processes that depend crucially on particular proteins with specific properties and functions. And based on function alone, we could actually classify proteins into 9 major classes. Many proteins are enzymes that serve as catalysts that greatly increase the rates of thousands of chemical reactions on which life depends. For example, the breakdown of food during digestion requires enzymatic function. On the other hand, structural proteins provide physical support and shape the cells and even organelles, giving them the characteristic appearance. Motility proteins play a key role in contraction and movement of cells and intracellular materials. For example, the actin and myosin are motility proteins that are found in your muscles. Regulatory proteins, on the other hand, are responsible for controlling and coordinating cellular function. This would include, for instance, the regulatory proteins that play a role in gene expression. Transport proteins are involved in the movement of other substances into and out of the cells or within the cells. Example of these transport proteins are, say, the channel proteins and even the carrier proteins are very classic example of transport proteins. Other classes of proteins would include signaling proteins that mediate communication between cells in an organism, and receptor proteins that enable cells to respond to chemical stimuli from their own environment. Finally, we have defensive proteins that provide protection against diseases, and the storage proteins that serves as reservoir of amino acids. Proteins are linear polymers of amino acids. Although more than 60 different kinds of amino acids are typically present in a cell, only 20 kinds are used in protein synthesis. Some proteins contain more than 20 different kinds of amino acids, but The additional ones usually are the results of modifications that occur after the protein has been synthesized. Although most proteins contain all or most of the 20 amino acids, the proportions vary greatly, and no two different proteins would have the same amino acid sequence. So this table shows some of the common small molecules in cells, like the amino acids, the sugars, lipids, and the aromatic bases. Now let's go back to the building blocks of proteins like the amino acids. Every amino acid has the same basic structure. Each have a unique side chain referred to as the R group. All amino acids except for glycine has an asymmetric alpha carbon atom. The specific properties of amino acids depend greatly on the nature of the R group. Every amino acid has the basic structure illustrated in this figure, with a carboxyl group and an amino group, a hydrogen atom, and a side chain known as the R group. All these are attached to a central carbon atom known as the alpha carbon. The R group is different for each amino acid and gives each amino acid its distinctive properties. With the exemption of glycine, for which the R group is just hydrogen atom, all amino acids have 4 different groups attached to the alpha carbon. Because the carboxyl and amino groups shown in this figure are common features of all amino acids, the specific properties of various amino acids vary depending on the chemical nature of the R group, which ranges from a single hydrogen atom in glycine to relatively complex aromatic groups, for example, found in tryptophan and tyrosine. The process of stringing individual amino acids together into a linear polymer involves a stepwise addition of each new amino acid to the growing chain by condensation or dehydration reaction. As each new peptide bond is formed by condensation, the growing chain of amino acid is lengthened by one amino acid. The peptide bond is illustrated schematically in this figure using a ball and stick models of the amino acid glycine and alanine. Peptide bonds have a partial double bond character, and thus the 6 nearest atoms are nearly planar. Notice that the chain of amino acids formed in this way has an intrinsic directionality because it always has an amino group at one end, referred to as the N-terminus, and it also has a carboxyl group at the other end, otherwise known as the C-terminus. This process of elongating a chain of amino acids is called protein synthesis. But the term is not entirely accurate because the immediate product of amino acid polymerization is not a protein but a polypeptide. A polypeptide does not become a protein until it has assumed a unique, stable, and three-dimensional state that is biologically active. Some proteins consist of a single polypeptide, and their final shape is due to folding and coiling that occurs spontaneously as the chain is being formed. Such proteins are called monomeric proteins. The word monomer literally means one part. Many other proteins are multimeric proteins consisting of 2 or more polypeptides that are often called polypeptide subunits. Although a polypeptide is itself a polymer, the entire polypeptide is sometimes monomeric unit of a multimeric protein. If a multimeric protein is composed of two polypeptides, it is referred to as a dimer. If it has three polypeptides, then we call it a trimer. This is hemoglobin. It's the one that carries oxygen in your bloodstream, and it is an example of a multimeric protein, known as a tetramer, because it is composed of four polypeptide subunits. There are two beta subunits and two alpha subunits. Together, it makes up this multimeric hemoglobin protein. Several kinds of bonds and interactions are important in protein folding as well as stability. The initial folding and subsequent stability of a polypeptide depend on covalent disulfide bonds as well as several kinds of non-covalent bonds and interactions such as hydrogen bonds, ionic bonds, van der Waals interaction, and hydrophobic interactions. Disruption of these interactions by heat, salt, chemical treatment can result in denaturation or unfolding of the polypeptide, making it therefore inactive and denatured in its unfolded state. Similarly, whenever there is incorrect folding of a polypeptide due to incorrect interactions, then serious biological effects can happen. In fact, the presence of misfolded proteins in cells can cause human diseases such as that found in Alzheimer's disease. Protein structure depends on amino acid sequence and its interactions. The overall shape and structure of a protein are usually described in terms of four levels of organization, each of these building on the previous one. There's the primary, secondary, tertiary, and quaternary. This table will show to you the different kinds of bonds and interactions involved in each of these levels of structure. The primary structure refers to the sequence of amino acids, while the higher levels of organization concerns the interactions between the amino acid residues. These interactions give the protein its characteristic conformation or three-dimensional arrangement of atoms in space. The secondary structure involves local interactions between amino acid residues that are close together along the chain. Tertiary structure results from long-distance interactions between stretches of amino acid residues from different parts of a polypeptide molecule. And when you talk about quaternary structure, it describes the interaction of two or more individual folded polypeptides to form a single multimeric proteins. All three of these higher level structures are dictated by the primary structure. The primary structure of a protein is a formal designation for the amino acid sequence. And when we describe the primary structure, we are simply specifying the order in which the amino acids would appear one after another. This is usually, by convention, always written from the N terminus going to the C terminus of the polypeptide. This is also the direction by which polypeptide synthesis would take place. The very first protein whose amino acid sequence was determined was the hormone insulin. This important technical advance was reported in 1953 by Frederick Sanger, who also received the Nobel Prize for this study or research in 1958. From his study, he was able to identify that the hormone insulin is made up of two polypeptides, the A subunit and the B subunit, with 21 and 30 amino acid residues respectively. The primary structure is very crucial. Genetically, it is significant because it is the sequence that is specified by the order of nucleotides in the corresponding messenger RNA. It is important structurally because the order and identity of amino acids would direct the formation of higher order structures like that of the secondary and tertiary structure. The secondary structure of a protein describes local regions of structure that results from hydrogen bonding between NH and CO groups along the polypeptide backbone. These results in two major patterns, the alpha helix and the beta sheets. The alpha helix structure was proposed in 1951 by Linus Pauling and Robert Quarry. An alpha helix is spiral in shape as you can see in this figure. It consists of a backbone of amino acids that are linked by peptide bonds with the specific R groups of the individual amino acids residues sticking out from it. In the alpha helix, there are 3.6 amino acids per turn, bringing the peptide bonds of the fourth amino acid in close proximity. The distance between these peptide bonds is, in fact, just right for the formation of a hydrogen bond between the NH group adjacent to one peptide bond and the CO group adjacent to the other, as seen in this figure. As a result, every peptide bond in the helix is hydrogen bonded through its CO group to the peptide bond immediately below it in the spiral, and through its NH group to the peptide bond just above it, even though the amino acids residues involved are not directly adjacent. These hydrogen bonds are all nearly parallel to the main axis of the helix, and therefore they tend to stabilize the spiral structure by holding successive turns of the helix together. In addition, two or more helices can coil together in a rope-like fashion to form a bundle of helices called a coiled coil, as you will see shortly in our examples later on. Please take note that amino acids that tend to form alpha helices would include leucine, methionine, and glutamate. It's also quite interesting to note that proline cannot form hydrogen bonds and therefore they have the tendency to disrupt alpha helix structures because they kind of introduce a kink or a bend in the structure. Another form of common secondary structure in proteins is the beta sheets. It's also initially proposed by Pauling and Corey. As seen in this figure, this structure is an extended sheet-like conformation with successive atoms in the polypeptide chain located at the peaks and throws of the pleats. The R groups of successive amino acids stick out on alternating sides of the sheet. Because the carbon atoms that make up the backbone of the polypeptide chain are successively located a little above and a little below the plane of the beta sheet. Such structures are sometimes called beta-pleated sheets. And note, just like the alpha helix, the beta sheet is characterized by a maximum of hydrogen bonding. In both cases, all the CO groups and NH groups adjacent to the peptide bonds are involved. However, hydrogen bonding in an alpha helix is invariably intramolecular, or in other words, within the same polypeptide. Whereas, when you talk about hydrogen bonding in beta sheets, it can either be intramolecular or it could be intermolecular, meaning linking two different polypeptides. The protein regions that form beta sheets can interact with each other in two different ways. If the two interacting regions run in the same N-terminus to C-terminus direction, The structure is called a parallel beta sheets. But if the two strands would run in an opposite N-terminus to C-terminus directions, the structure is called antiparallel beta sheets. In some proteins, several antiparallel beta sheets associate symmetrically around a central axis in a structure that is known as a beta propeller. The tertiary structure reflects the unique aspect of the amino acid sequence because it depends on interactions of the R group. Tertiary structure is neither repetitive nor easy to predict. It results from the sum of hydrophobic residues avoiding water, hydrophilic residues interacting with water, the repulsion of similarly charged residues, as well as the attraction between oppositely charged residues. The most stable possible three-dimensional structure of a particular polypeptide is what you call as native conformation. And proteins can be divided into two broad categories: fibrous proteins or globular proteins. In the case of fibrous proteins, they have extensive regions of secondary structures. This is an example of a fibrous protein, particularly fibroin. It consists mainly of long stretches of antiparallel beta sheets, with the polypeptide chains running parallel to the axis of the silk fiber, but in opposite directions. The result is a silk fiber that is strong and relatively inextensible because the polypeptide chains in a beta sheet conformation are already stretched to nearly their maximum possible length. Hair and wool fibers are made of protein alpha-keratin, which is almost entirely alpha-helical and is stabilized by hydrogen. In forming the hair, two alpha helices are coiled around each other, as shown here in this diagram. And two of these coils would pair to form a protofilament containing four alpha helices. And groups of 8 protofilaments will then interact to form an intermediate filament, which would bundle together to form the actual hair fiber. Not surprisingly, the alpha-keratin polypeptides in hair are rich in hydrophobic residues that interact with each other where the helices would touch. This allows the tight packing of the filaments of hair. Globular proteins is another example of a protein. These are proteins that are folded into compact structures. Each type of globular protein has its own unique tertiary structure. Example of which would be enzymes. Most enzymes are actually globular in nature. The enzymatic function of an enzyme depends critically on their proper structure. This figure shows the native tertiary structure of your ribonuclease enzyme, which is a typical globular protein. This shows two different conventions that can be used to represent the structure of ribonuclease. Above, you see the ball and stick model here. And the spiral and ribbon model found below. For clarity, most of the side chains of ribonuclease have not been shown or are not shown in these models. The groups in gold found in the figure above are the 4 disulfide bonds that would help stabilize the tertiary structure of the ribonuclease. So here's one. And here's another one. And here's the third one. And the fourth one. Globular proteins can be mainly alpha helical, mainly beta sheet, or a mixture of both structures. Globular proteins can be mainly alpha helical. Example, the coat of the tobacco mosaic virus in figure A here. Or it could be mainly beta sheets, such as a portion of the immunoglobulin antibody. Or they could be a mixture of both. In the case of the beta sheet, it appears like a barrel configuration. For the third example, the hexokinase, it shows a twisted sheet which is a combination of both alpha helix and beta sheet. Please take note that many globular proteins are actually consist of a number of segments that are called domains. In fact, in figure B here, what is being shown is only a portion of the immunoglobulin, particularly the V2 domain of the immunoglobulin. In figure C, what you see is a portion of the hexokinase enzyme, particularly the domain 2 of hexokinase. Many globular proteins consist of a number of segments, as we mentioned earlier, called a domain. A domain is a discrete, locally folded unit of a tertiary structure that usually has a specific function. Each domain may include as little as 50 to as many as 350 amino acids, with regions of alpha helices and beta sheets that are packed together compactly. Small globular proteins are usually folded into a single domain like the ribonuclease as you've seen earlier. In this particular example, you can see a protein that is composed of a single polypeptide that is folded, showing two of its functional domains. Domain 1 here and domain 2 here. It is known that the primary structure determines the final folded shape of a protein. However, we are still not able to predict exactly how a given protein will fold, especially for very large proteins. This brings us to the quaternary structure of a protein. This is the level of organization concerned with subunits interaction as well as assembly. The term applies specifically to multimeric proteins. Some proteins consist of multiple identical subunits, for example, hemoglobin, that is made up of 2 alpha subunits and 2 beta subunits. The bonds and forces that maintain quaternary structure are actually the same as those responsible for maintaining the tertiary structure. And the process of subunit formation is usually spontaneous. More often, molecular chaperones are required to assist in the process. And when I mean process, I'm talking about proper interactions and folding. A higher level of assembly is possible in the case of proteins, like say enzymes. These are organized into multi-protein complexes, and each protein in the complex may be involved subsequently in a common multi-step process. An example is the pyruvate dehydrogenase complex, in which 3 enzymes and 5 other proteins form a multi-enzyme complex. This ends part 1 of our lecture on macromolecules of the cell. Do check out the video for the part 2 of macromolecules of the cell, this time focusing on the role of proteins in diseases, particularly Alzheimer's disease. Thank you for watching.